Today we're diving into the assembly of two closely related electrochemical cells, the bottom-mounted two-compartment cell and the bottom-mounted H cell. Starting with the working electrode mounting process, which is identical for both cells, begin by twisting off the glass chamber and placing it to the side. Then, go ahead and unscrew all 4 nylon screws. Holding the two halves together, gently place them on a flat surface and lift the top half away. Now you're ready to insert your electrode. In our demonstration, we'll be using a piece of titanium plate. It's important to align the plate's edges with the screws to ensure that it is centered. Once that's done, carefully position the top half back on, making sure that it lines up with the pins. To secure everything in place, retighten the screws in a cross pattern. Do ensure you tighten them evenly so that there is a consistent gap between the cell halves. Move on to the lid for the two-compartment cell. Start by removing the inner compartment with a gentle twist and pull. Next, fit the counter electrode into the lid, and using tweezers, push the plug in to ensure an airtight seal. With that in place, you can then return the glass compartment into its position. Now for the reassembly. Twist and push the glass body onto the base, followed by the lid. For the reference electrode, remove one of the plugs and insert an O-ring with the help of your tweezers. Ensure the O-ring settles into the inset. Now, uncap the electrode and give it a gentle tap to remove any trapped air bubbles. With care, twist and insert the electrode into the cell. Remember, it might be a snug fit, so it's essential to avoid damaging the electrode. The reference electrode should be placed a few millimeters away from the working electrode. Voila! Your cell is now primed and ready for your scientific explorations. Let's now shift gears to the bottom mount H cell. Since the working electrode assembly remains the same, let's focus on the lid. Grab the second lid and position the counter electrode, pushing down firmly with your thumbs to guarantee a tight seal. Then, return the second lid back into the primary one. If there's ever a need to swap or change the membrane, simply unscrew it, lift out the membrane holder and replace as needed. When you're screwing it back, ensure that the plate aligns parallel to the body. Now, twist the lid onto the glass body, remove the plug, add an O-ring, and then introduce the reference electrode just like we did for the previous cell. And there you have it. This cell too is all set up for your experiments. Should you have any queries about our electrochemical cells, please don't hesitate to visit our website. Happy experimenting!